place that clinically we're trying to make the biggest impact is in the domain of musculoskeletal diseases like arthritis, tendinopathies, bone fractures. All of those tissues, the tendon, the cartilage, the bone, ligaments, they're all made up of the same protein, type 1 collagen. We do a lot of work on trying to study the way it self-assembles to try to figure out how those diseases start and then evolve into the things that give us problems. If, for example, you come to the doctor and you're complaining that your knee hurts because you have runner's knee, usually they can look at it non-invasively with something like MRI. They see that there's a lot of damage. There's a lot of extra water in the tissue. There's a lot of swelling. These tissues, they have the ability to repair, but it's very limited. And so if you wait until the disease gets too far along, then your body can't repair it anymore. So our goal is to try to look early on at a small length scale, at the nanoscale, to see how the disease is starting. With this idea of tendinopathy, we want to understand how the individual collagen molecules that make up your tendons end up self-assembling when they're produced by the cells that make your tendon. If you could shrink yourself down to being really, really small and look around at what's going on when your tendons are assembling, what you would see is a lot of motion. The collagen molecules are like little cooked spaghetti noodles. They're oscillating all over the place. They form a really nice crystal, this long cylinder called our collagen fibril. We would like to understand what are the specific forces of interaction that cause these collagen molecules to interact with each other. I have a patent on a bone substitute material. It duplicates the structure of bone at a length scale below a micron. And we think that utilizing these ideas of self-assembly of collagen and then using the collagen as a template to mineralize that structure, that we can make a material that will bear load. You'll be able, for example, to put it in your jaw and then chew, where this material is actually providing the strength and the stiffness to allow that to happen. We're also taking these ideas of self-assembly and applying them to trying to make an artificial ligament, artificial tendon. We're trying to take collagen molecules, trying to attach these things onto a titanium surface. Titanium is the favored material that orthopedic surgeons like to use when they put it in your bone. And then use those individual molecules to grow the fibril around them. So we get like telephone poles of collagen coming off of the titanium surface. And we're hoping that those will integrate into the bone in a biomimetic fashion.